Hello, thank you for attending today's session. My name is Miguel Tam. I'm a strategic marketing director at BioLegend, and today I wanted to go with you over some of the solutions that BioLegend has on flow cytometry and multiplex assays. This is our agenda, and I will go over some of the flow cytometry reagents that we have, the type of fluorophores and the applications that we cover, as well as the multiplex solutions that we have to detect soluble molecules. I'm going to go over the workflow and then our portfolio for that particular set of reagents that we offer. And first, I wanted to go quickly over the basic principles of flow cytometry. Here to the left, we have this diagram showing how different cell types can be detected using flow cytometry. Typically, there is a laser that is going to excite a fluorophore that is conjugated to an antibody typically, and that antibody is going to detect molecules that are expressed by these cells. Then the signal that is collected from that excitation is processed by this instrument, and you can see here just simple examples of expression of CD4 and CD8, for example, in T cells. However, the workflow for flow cytometry is really dependent on conjugates that are going to be used to detect these cells. And as you can see here on the plot to the right, until very recently, instruments were offering a number of parameters to analyze in cells, up to 18, as you can see here in this graph back in 2010. However, the availability of fluorophores to detect molecules was always ahead of the number of parameters that were offered a few years ago. As you can see here, there were more than 40 different fluorescent molecules that were offered back in 2010, as compared to 18 different parameters that were offered by the instruments. And here at BioLegend, we really specialized in conjugating fluorescent molecules to antibodies that can be used to detect cells. And in the next slide here, I'm showing you how this process is evolving, because new technologies such as mass cytometry and spectral cytometry are providing more and more channels or parameters to be used to detect proteins in the surface or in the intracellular space of the cells. And here at BioLegend, we have a large collection of conjugates that people can use to detect molecules expressed by these cells. You can see here that we have more than 40 different molecules that can be conjugated to antibodies, and the number of conjugates that we have by molecule, starting with the more popular ones like PE, going down to less used molecules, but we do offer a very wide selection of different conjugates that can be used to analyze cells. But if we dive a little bit deeper into the structure of these molecules, we can see that we have simple organic dyes, such as FITC and Alexafluor 647. You can see the structure here. And we are expanding this portfolio with our proprietary Spark dyes. And we are also expanding our portfolio with with fluorophores that are based on proteins, such as PE, APC, or PerCP. Our tandems based on these proteins are called FIRE. And in addition to that, we also have an expanding or growing portfolio of fluorescent complex molecules, such as the brilliant violet dyes and the Kitevia dyes. We can see here that we can apply these reagents in multiple applications, such as immunophenotyping, cell sorting, and also to study cell physiology. We cover a big number of cell surface and intracellular targets, and in cell physiology, we can look at cell cycle, DNA content, and cell proliferation. These tools can also be used for characterizing the immune response and to characterize the dynamics of the cell populations, also to quantify cells, to look at cell signaling and also to look at cell-specific cytokine secretion. In addition to that, they can also be used as diagnostics, and we're going to be hearing a lot more about diagnostics and cytokine detection in this talk, because cytokines are very important in mediating the immune response. And this slide here is just to show you how different cytokines can be mediating different type of responses. And measuring these cytokines is critical to understand these immune responses. And one tool that we have developed to measure cytokines very efficiently is a multiplex assay that is made of beads that are conjugated to captured antibodies. These captured antibodies will bind to soluble molecules that are present in a sample, and then we will use a bioaccumulated detection antibody that can bind to that molecule as is described here. And the bioaccumulated detection antibody then binds to a streptavidin PE conjugate. That PE signal then is measured on a flow cytometer, and the amount of analyte that is present in the sample is directly proportional to the PE signal that is detected in this assay, as you can see here in this example on that, it's in this standard curve. Now, to just explain a little bit more our multiplex solution, we call it LEGENDplex. You can see a picture here of this kit. Our multiplex assay is compatible with existing instruments, and they provide an excellent value. And they also come, it also comes with the free analysis software. We have a wide portfolio covering human, mouse, rat, or non-human primates. We have more than 50 predefined panels, plus we have individual reagents. It allows for great flexibility because we can design custom panels, and you can multiplex up to 14 different analytes in one single assay. And you only need 25 microliters of sample to measure all these 14 analytes. And to give you an example of where these assays are really making a difference, I wanted to show you a workflow that many researchers in the community are doing right now to study COVID-19. And it all starts with the viral recognition and the inflammation that is caused by the virus. And there is a cellular response against that virus. And there are biomarkers that need to be monitored during this immune response. And with that, I wanted then to introduce Dr. Bruce Patterson, who is going to explain how he is analyzing samples from COVID-19 patients and using LEGENDplex and flow cytometry as one of the tools to study these patients. Thank you.
Welcome. I'm Dr. Bruce Patterson. I'm the CEO of InCellDX, Inc. I'm going to talk today about the diagnosis, treatment, and immunopathogenesis of chronic COVID-19, or PASC. I'd like to thank BioLegend for sponsoring this talk, but for also being a tremendous partner in what you're going to see today. From the beginnings of COVID-19, we have been able to describe the pathogenesis of COVID in phases, with the initial viral replication phase, and then eventually the immune system kicks in, and you have early, moderate, and severe inflammatory reactions that some describe as the cytokine storm. What we found, which is very interesting, is then you have a cytokine flip following recovery from acute COVID-19. And what we noticed at the end of several clinical trials last spring was that patients were going home after being hospitalized and recovering from their illness, and even saving some of them from intubation and ECMO. And what we found is they were alive, they were better, we were sending them home, but by no stretch of the imagination were they immunologically normal. If you did a comprehensive analysis of their immune profile, you would notice that they were far from normal in terms of both their immune cellular levels and subsets, and also far from normal in terms of their inflammatory markers. And initially, we studied over 100 biomarkers in COVID in the early days, in the February 2020 timeframe. And what we did is we really came up with a panel, which I'll be describing, that constitutes a number of very important inflammatory markers that transcend both acute COVID infection and also what we now call long haulers or PASC. We did this and created a kit called InCellDX in collaboration with BioLegend. And one of the most important aspects in terms of doing immune profiling, especially when it involves cytokines, was to have reproducibility and clinical accuracy in a sample that can be quite labile. And again, I think the quality of this product manufactured by BioLegend really helped us to bring it to the clinical marketplace where it could be best utilized. It consists of 14 cytokines and chemokines directly involved in COVID-19. There's been other scores developed to look at severity of COVID using cytokine panels. And we developed our own, as you will see in a moment. And what this does is it really enables or can enable practitioners to treat with precision the impacts of COVID-19 in all patients, including long haulers. In my almost 30 years of viral pathology research, I've never seen a complicated disease such as SARS-CoV and COVID-19. And because of that, it demands the use of precision medicine like we use in cancer. And I think that's been one of the drawbacks so far of what I've seen in the pandemic, having been now through 4 pandemics and starting my research in HIV, is a lack of using all the tools that we have at our disposal to be able to not only diagnose patients, but also to triage patients to therapy, and most importantly, save lives and manage precious resources. This panel, which can be done on any flow cytometer, does the 14 cytokines and chemokines at the same time in the same well. So it's very scalable and high throughput. And that's been critical when you think about the numbers of samples. If you think about long haulers alone, with the current estimates being 10 to 30% of all individuals who had active COVID, you're talking about enormous numbers of patients who are suffering and have significant disability due to the long-term effects of COVID. And that's our target audience in terms of deploying precision diagnosis and precision diagnostics and using that to both institute therapy and to monitor therapy and to monitor recovery in a non-subjective manner in a condition such as long haulers, which some reports say may have as many as 200 symptoms associated with that. And in the 5,000 patients that have come in contact with InCellDX for this testing, we found every patient has their own story. Every patient has their own level of disability. Every patient wants to get back to normal. And the only way to do that is through precision medicine. This is just a few of the long hauler symptoms that we reported in our first paper, including fatigue, brain fog, shortness of breath, peripheral neuropathies, tachycardia, amongst others. Again, this is by no means a comprehensive list, but as we went through our first 160 patients and published on those, we really found that these were the most common symptoms that we came across. Our paper, Immune-Based Prediction of COVID-19 Severity and Chronicity Decoded Using Machine Learning, was a paper that we wrote in the middle of last fall. Long hauler patients had barely been characterized in this time frame, but based on our findings at the end of the spring from the clinical trials showing that individuals did not have a normal immune response or a normal immune profile after active COVID, it was very important to us to say, what are the differences between someone with active COVID and somebody who has these long hauler symptoms? So we deployed machine learning and AI to help us go through all of these hundreds of biomarkers to figure out, were there differences? And do those differences demand different treatments? And the answer to both of those questions was yes. And, you know, it's come to just the last month or so, maybe 2 months, that the world has started to recognize long COVID. And in fact, we understood the immunology of long COVID way back in the fall, and have since gone on to use precision medicine to start treating these individuals. When you look at the immunologic profile of these individuals, and again, I apologize for these numbers being very small, but there was a lot of parameters that we were looking at. On the top row, we looked at the cellular constituents. We looked at cellular subsets of T cells, B cells, monocytes, macrophages. We looked at markers of exhaustion like PD-1, LAG-3, CTLA-4. We looked at the levels of T regulatory cells, which shut off the immune system in these individuals. And some of the profound findings that we saw in long COVID was that 25% of long haulers still months after active COVID had low CD8 percentages. That was very compelling. The other important finding was, and this was across the board in 100% of long haulers, was this increase in various monocytic subpopulations. And that is something that I'll talk about in greater detail shortly. But what we found as you look at monocytic subpopulations, classical monocytes, CD14 high, CD16 negative, intermediate monocytes, CD14 positive, CD16 positive, and the non-classical monocytes, CD14 low, CD16 positive. All of these subpopulations of monocytes have different functions. They express different cell surface receptors, in particular the ACE2 receptor, where classical monocytes express far more ACE2 than non-classical and intermediate monocytes. And they also express different levels of CCR5, which is this receptor that allows them to migrate throughout the body in response to the production of CCL5 or RANTES, much like bees are attracted to honey. Along the way, we also found significant cytokine abnormalities. And again, this was using this kit that we developed in collaboration with BioLegend that turned out to be extremely reproducible, transferable, disseminatable, all the great words that you use in the clinical realm as opposed to life sciences and research. And we're now fully live at several reference laboratories, and we're seeing the advantages of having an assay that is non-subjective, that can tell us whether or not the immune profile that we see in long haulers is improving with therapy. And classically, as you can see in the lower row, long haulers, unlike active COVID, had elevated levels of IL-2, interferon gamma, high levels of CCL5, high levels of TNF alpha, and very interestingly, very high levels of VEGF and soluble CD40 ligand, which has led us to believe and led us to suggest in our latest paper that vascular inflammation is one of the hallmarks of long haulers and one of the keys to addressing in terms of therapy of long haulers. When we applied machine learning to the cytokine panel that we developed, the InCellDX panel in 14-plex, we found some very important patterns. Number one, we found a signature for long haulers, or PASC, that was based on feature importances identified during machine learning. And we found that the long hauler index, consisting of interferon gamma plus interleukin 2 divided by CCL4, divided in the high 90%, 96, 98%, divided long haulers from active COVID and from normal individuals. And to us, that was both a surprise and also a very important finding in terms of finding some commonality in long haulers amongst great heterogeneity, both in symptoms and other cytokines that are expressed. Similarly, our analysis identified a severity score in those individuals with different severities of active COVID. So we looked at mild to moderate patients, normal patients, of course, severe patients, critical patients. And we found a severity index shown in the formula above, which is interleukin 6 plus soluble CD40 ligand divided by 1,000 plus VEGF divided by 10 plus 10 times IL-10 divided by IL-2 plus IL-8, which clearly distinguished severe COVID from mild to moderate COVID. And we think this formula and this panel of cytokines can clearly delineate and help us triage patients who are going to go on to severe disease in an effort to, once again, save lives, treat appropriately, and triage individuals so that we can maximize the utility of our resources around the world. We use this data to establish clinical cutoffs for the long hauler index. In the upper left, you can see normals in green, well below the dotted line or cutoff for those being long haulers. Mild to moderate in blue, again, below the dotted line. And the severes below the dotted line as well, except interestingly for, I believe it was 5 patients out of the 50 or so that we studied that actually started to approach registering on the long hauler index. Now, we posed the question in the discussion of our new paper, are these individuals that are already developing into long haulers even while at the tail end of their severe disease? We all know that long hauler syndrome or condition comes in individuals who have mild disease, moderate disease, and severe disease. But we found it very important and very interesting that some severe patients, say at 28 or 48 days, were transitioning into an immune profile that resembled long haulers, which you see in the right in yellow, which are well above the dotted line cutoff. Here you see a composite showing very clearly the distinction between long haulers in yellow, severe cases in green dots, mild to moderates and normals in blue, in light blue and dark blue, below the two dotted line cutoffs, so that we could clearly delineate using a multi-disease classifier, mild to moderate from severe from long haulers, just based on their immune profile. And again, I think this tool has been incredibly important since we began using it in the fall to really say, you know, what is the severity of your acute disease? And are you a long hauler, which I think is half the battle, identifying that these individuals who sometimes are ignored or thought to have other conditions actually have something very real. And they have something that is causing great disability, and they need to be recognized. And I'm very pleased to see that notion has been raised all the way up to Congress, who is trying to pass bills for these individuals suffering from long COVID and providing benefits. Again, this is a panel, again, very small, but I wanted to point out that we've already started to go to the next level, which is to establish cytokine signatures for all the individual symptoms. We call it symptom-specific cytokines, or SSCs, where we know the cutoffs and the ranges of values of our 14-plex that are associated with different symptoms, so that when we think about treatment, we can tailor our treatment so that we can address the individual symptoms. And the other piece of this puzzle, which is really a triad, identify long haulers, which was very important. Number two, identify which symptoms are associated with which immunologic abnormalities. And last, which therapies address the immunologic abnormalities that contribute to the symptoms. If we can connect those three dots, then I think we're well on our way to providing effective therapy against long haulers. Some of the important publications that we've put out to show what we think is causing long haulers was first in a paper published in November, where we saw viral RNA shedding in an individual with low CD8% for more than 87 days, or close to the 3 months, which is the cutoff for long hauler conditions. And what was really interesting is 25% of long haulers, months and months and months after acute COVID, still have low CD8 percentages. And again, that seems to be a factor that can contribute to viral persistence. What we also found is that there is a cell type, which I will show you, but this is actually the profile of several long haulers that we've treated with the CCR5 antagonist Maraviroc. Now, Maraviroc has a number of different activities that I believe are important to the treatment of long haulers. Number one, it brings down RANTES, which is elevated in long haulers as well as active COVID. And it brings it down, and again, that it's important because of the role in migration and obviously chemotaxis of immune cells throughout the bodies, causing immune activation in those particular sites of the body. Now, It also brings down interleukin-6, as we published last year. It also, very importantly, brings down VEGF. So it seems to have some ability to relieve vascular inflammation. And in fact, the decrease in VEGF is associated with improvements in brain fog and in peripheral neuropathy, which VEGF has been attributed to. And so it has this multitude of effects that have been positive for those with long hauler symptoms. And again, using this very simple procedure, I won't go into great detail, has allowed us to do reproducible serial sampling of these individuals, like I showed in the previous slide clinically, to monitor their response. So these two individuals that we showed here had multiple samplings, as you can see in the top example. There's two separate serial cytokine panels. The long hauler index decreased. The patient's symptoms improved significantly, and that was reflected in the number of different cytokines and chemokines that were reduced by CCR5 antagonism. Another important aspect is that this assay uses standardized CPT codes. I won't go into that in great detail, but one of the most important aspects of long haulers is they're disabled. A lot of them are out of work. They are desperate to feel better. They are desperate to get back to work. And we have to be cognizant of the economic hardship that they're going through. And that's why, in parallel to what we're doing from a precision medicine standpoint in treating these individuals, we're also pushing for legislation to help pay and provide benefits to get these individuals better. This is a report done from one of our reference laboratories of the 14-plex panel. Again, I think they've been impressed with the reproducibility. They've been impressed with the ease of validating this under CLIA. So kudos to BioLegend for their great manufacturing practices to allow us to provide a kit tailored for the number one, the severity index and the long hauler index that I described prior. Now, the most important aspect of all this work that has been brought together through immune profiling is that we actually found long-lived cell types still containing SARS-CoV protein in long haulers up to a year post-COVID diagnosis. This is an individual that just celebrated, and I use that word loosely, their 1-year anniversary of being infected with COVID. We did a profile using a SARS-CoV-2 antibody in addition to our monocytic subset analysis, and we identified the three populations of monocytes: the CD14 high, CD16 negative population, the CD14, CD16 dual population, the CD14 low, CD16 positive population. And then we asked the question, do any of these scavengers or antigen presenting cells still contain remnants of SARS-CoV-2? And indeed, we found it. And we found it in the CD14 low, CD16 positive population. As you can see down here, almost a log separation from the other monocytic populations of this cell type containing SARS-CoV-2 protein. We think this is providing constant antigenic stimulus to the immune system. And In fact, this cell type has the ability to migrate throughout the body in response to CCR5. It expresses CCR5. It also expresses the receptor for fractalkine, which is expressed on vascular endothelium. So we think that ties together this narrative that we've discovered in terms of the cytokine immune profile, in terms of the ongoing inflammation seen in long COVID. And in fact, what's very interesting is all the long haulers that I've spoken to have exercise intolerance. This tends to wax and wane. They feel better, and they go on and they go and exercise, and they're bedridden for 3 days or even a week. And we think that's because of the mobilization of this particular cell type. Now, part and parcel to, obviously, CCR5 therapy, as we published last year, is the importance of knowing whether or not the CCR5 inhibitors are actually bound to the receptors and to what extent. Way back in the mid-90s, we were in the early days of discovering that there was two different types of CCR5. There was wild type CCR5 and delta 32, an allele with a 32 base pair deletion that conferred protection against HIV infection. And what that really led to was an assay called receptor occupancy. There's two separate assays for CCR5 in terms of determining its effects in HIV, in cancer, and in COVID. In HIV, receptor occupancy is critical to show that the drug is blocking the receptor that HIV is using. There's another assay called tropism, which is solely predicting that the virus actually infecting an individual uses CCR5. So in other words, tropism is about the virus, receptor occupancy is about the cell. By no means does our receptor occupancy ever predict that a virus will bind to CCR5. Both assays are needed, at least in the HIV context. In cancer and in COVID, it was more important to see what the levels of CCR5 were on particular immune cell subsets because it varies. We published a paper in the mid-90s showing cytokine regulation of CCR5. So the difficulty in using CCR5 antagonists is the fact that CCR5 is a moving target. So receptor occupancy is essential. But what is really essential is having a pattern you see like this. The blue line and the green line are CCR5 clones that are not blocked by drug. That is your internal control because the CCR5 levels differ depending on the time, depending on inflammation, depending on infection. The red line and yellow line, conversely, reflect serial increases in drug concentration and inhibition of antibody binding to CCR5. The combination of a non-blocked antibody and a blocked antibody and looking at that ratio is critical in COVID, HIV, and cancer, where CCR5 is becoming extremely important in terms of being a new immuno-oncology target. And in fact, BioLegend helped us label CCR5 in such a way that it had a 1-to-1 F to P ratio. In other words, we knew one fluorochrome equaled one receptor. So we could calculate exactly the number of receptors on each of these cell types, the CD14 monocytes, the CD14, CD16 macrophages, the effector memory T cells, CD4, CD45RO, and of course the Treg cells, the CD4 FOXP3 T cells. This was a critical reagent to help us figure out the correct dosing. Receptor occupancy is all about dosing. and has nothing to do with any virus or anything other than what dose do we use so that we inhibit the Treg cells from shutting off the immune system, but don't completely inhibit the effector T cells that are needed to fight off either COVID or virally infected cells or cancer. I'll finish that with that, but in fact, we can apply the same technology with the same antibody manufactured by us and labeled by BioLegend to do CCR5 expression using without antigen retrieval, so that we can see in cancers the level of CCR5 expression as this comes to bear in terms of being an immuno-oncology drug to inhibit Tregs and repolarize macrophages from a pro-inflammatory, pro-tumor phenotype to an effector macrophage phenotype. And again, we see beautiful staining in blue with this conjugate from BioLegend labeled with a fluorochrome that we can then stain for in tissue without the need for antigen retrieval. So it's truly, truly quantitative, both at the cellular level and on the tissue level. So I'll end there basically with, some parting words that CCR5 antagonism we've shown in COVID and in long haulers is critical to therapy. I also showed data where it's important for cancer in determining who will respond to therapy. And InCellDX has developed an app called ImmunoTrack that we can incorporate immune symptoms, immune values with blood pressure, body temperature, heart rate, blood oxygen, and heart rate variability to show the effects of anti-inflammatory and immune modulation on patients, where they can monitor their symptoms on a day-to-day basis and contribute holistically to our assessment in a precision medicine basis. I thank you for your time. I am so grateful to BioLegend for making great components and raw materials for InCellDX for our unique applications. And with that, I'll end, and I'm happy to answer any questions. Thank you very much.